No, sorry, agree to disagree. Captain Picard is obviously not only the better captain, but also the better looking captain than Captain Kirk. Oh, sorry, I have lecture, gotta go. Sorry about that, folks. So I'm hoping that you've also had a little bit of time to relax and enjoy yourselves. Again, hoping that neurophysiology is something that's interesting and fun for you, and that you're also making time for yourself. Your mental health and well-being are just as important as doing well in this course. And taking breaks once in a while is actually a really good thing and a positive thing. So I'm hoping that you have started to schedule that time for yourselves a little bit as well. So today we're going to be talking about sensory systems. Now, every single part of today's lecture, I want you to remember, apply to the sensory nervous system. We're going to get into that in a little bit more detail later on, but I wanted us to start thinking about how we can adapt the terminology and everything that we've learned so far in what we are talking about specifically with the sensory nervous system. So I'm going to go ahead and share my screen with you. And we'll get going in a second. All right, here we are. And again, in case you weren't aware, and if you're wondering who I am, this is me. You're always welcome to email me if you have any questions, if you wanted to talk about Star Trek, or you wanted to tell me how you're doing and just about your well-being or different plans that you are taking time off and doing. I'd really love to hear from you. Today, we're going to be talking about special cases, about the action potentials, receptor potentials, and we're going to be looking at the sensory system. And everything in today's lecture, in episode number 5, deals with the sensory system. And again, I'm using the notes that are adopted and adapted by my colleague, Dr. Nojin Key. So let's just jump right in. So we're going to be looking at doing the same things that we did in the previous lecture, in lecture number 4, getting a signal from the outside environment. But here, instead of looking at postsynaptic potentials, we're going to be looking at the parallel within the sensory nervous system. So we're making a little bit of a leap, but you're going to see why it's important a little bit later on. And these receptor potentials use the same mechanisms. We need receptor proteins. So just like postsynaptic potentials, we need receptor proteins. We need to get that to the next part of the nervous system. So we have to transmit that signal, just like the spread of that postsynaptic potential. And then we're going to look at some very interesting phenomenon about how these sensory nervous system components can signal for stimulus intensity. So how does your skin know if it's feeling something soft or if it's feeling something like much more pressure on your skin? And how do we actually code for modality? And I'll give you a specific example of modality coding a little bit later on, and some controversies about that field. And again, it's really, really cool. So, and we'll end off with perceptive fields, which will set up for the sensory nervous system when we come back from break starting next week. So we're almost there, folks. Bear with me. So here, what we're looking at is what we call a receptor potential. And a receptor potential is a change in the membrane potential of the cell that is responding to a sensory cue that's coming from outside of the body. And this is an exterior sensory cue. So energy from the environment, it could be a chemical signal, or it could be light, it could be sound waves. The energy in the environment will react with membrane proteins, just like we looked at in the last lecture in episode number 4. And in general, this causes depolarization of the cell that's responding to it. Okay, so very similar to what we looked at before. And depolarization of sensory receptors with specific forms of energy that they are aligned to. So just like glutamate binds to glutamate receptors, we can't have light interacting with receptors that would normally respond to sound, etc. We'll get to that a little bit later, but they are very specific. And generally, we're talking about depolarization. And the exception, when we get into the vision lecture section, are these photoreceptors that will actually hyperpolarize under different light stimulating conditions. All right, so let's go into this receptor potential. As a parallel to the postsynaptic potential that we talked about in the last lecture. When did you watch that last lecture? I have no idea, but I hope that it wasn't too long ago. I'm also hoping that you took a break from that last lecture before coming to this one. Similar to the postsynaptic potential that we talked about, the receptor potentials are also encoded or are responsive because of these receptor proteins that are embedded in the cell membrane of these sensory neurons or these sensory systems. So the receptor proteins in these sensory cells also change shape when specific energy, whether it's chemical energy or whether it's physical energy in the form of sound waves or photons of light, whatever that energy is, they will change shape. And in many ways, it parallels what we looked at with ionotropic and metabotropic systems as well. They could directly open an ion channel, and it could be a cation channel that leads to depolarization, so almost identical to ionotropic receptors. So I'm hoping that parallel is made for you, or it could respond using metabotropic systems. Again, very, very similar and a parallel to what we looked at in postsynaptic cells. Here we're looking at sensory cells. Remember, this whole lecture is devoted to sensory systems and sensory cells. So for these postsynaptic receptors, a chemical stimulus, for example, the ones that we encounter most often, binds to a specific G protein or metabotropic receptor. It activates the G protein as it changes shape of the receptor. It activates a G protein. This will then activate adjacent enzymes like adenylate cyclase or adenyl cyclase. This in turn will produce second messengers like cyclic AMP. And cyclic AMP, as we learned in the last lecture, will turn on protein kinases. And these protein kinases can then in turn activate or phosphorylate ion channels. And in the specific example of the beta adrenergic receptor, we looked at the phosphorylation of calcium channels, keeping them open longer. Hoping that you remember that because it's going to be important a little bit later on. And you can read into that however much you want to. So in terms of the stages of amplification, sorry, I'm going the wrong way. In terms of the stages of amplification, there are two of these stages. So these G proteins can activate a number of different enzyme molecules, and each of these enzyme molecules in turn will produce lots of the second messenger system, like more and more of the cyclic AMP. So a single stimulus molecule, and I'll show you that on the next slide, will be able to produce many, many-fold increases in the second messenger. So here's an example of what I mean by that. And this is very commonly used throughout the sensory nervous system. And here's an example of the sensory nervous system where we're looking at the sense of smell. And it's a very, very sensitive type of sense. So this olfactory receptor that is here embedded on the plasma membrane. So here is the plasma membrane and would normally be encoded or found in this region identified here. These specific receptor proteins that you see embedded for an odorant, and that's a chemical signal, these specific receptor proteins will bind to specific odorants. So not all odorants bind to all or one receptors. We have individual receptors for each. In turn, after this has bound, it will activate the G protein, which will then activate the adenylate cyclase. And in turn, it will then convert ATP to cyclic AMP. And this cyclic AMP, much as we saw in the last lecture with beta adrenergic receptors, will then phosphorylate the calcium, the sodium calcium channel that you see here. So this is a little bit less specific. So it's not just specific for sodium. It actually lets in both sodium and calcium, and it will actually activate it so that it stays open longer as long as that odorant is present. And in turn, as calcium and sodium are entering, this will cause depolarization of this membrane or this cell. So we still get that depolarization event. And hopefully now things are kind of clicking into place and some of this makes a little bit more sense to you. Now, why, again, is that important? So similar to the parallel that we tried to draw up a little bit earlier on, this depolarizing current that we just saw being generated, so we see a change in the membrane potential, actually has to travel down toward the trigger zone of the actual axon itself. So this is a very specific scenario. So we've stimulated and the odorants are binding here. We need enough of this depolarizing current to actually work its way a fair distance down this particular cell toward the trigger zone. So the concepts and this idea of a trigger zone, and you hopefully know by now because we've talked about it in almost every single lecture, this is where we see the voltage dependent sodium channels and this is where we have excitable membranes, we need to have it above threshold by the time it reaches here. So the same concepts just being applied to the sensory system. So this is an example where the sensory cell, in response to a specific mode of energy, here a chemical form of energy, actually will generate an action potential at this spike generating zone. So the signal can actually create a change in ion movement, and that's what we just saw in that previous example. A second way in which sensory cells can actually transmit signals is through not just these action potentials, and I'll get into this in a little bit more detail. And that previous example of the olfactory system is one. So if we look at these axon terminals, especially in sensory axons that innervate different parts of the skin, the first patch of the excitable membrane will generally be at these branch points. So this is where we would expect to see it. On that previous example in the olfactory system, we saw it at a distance from where the olfactory signals were generated. But here we also see the receptor proteins are embedded within the membrane. This is where the depolarization begins, and the trigger zone is at a distance. So these receptor potentials, so we have this receptor potential. So think of it as a specific form of a postsynaptic potential. It's not being generated by a neurotransmitter. It's being generated by a sensory cue or a modality of energy. And this could just be like pressure on the skin. It will activate these receptors, will cause depolarization. And as a result, if these depolarizations are at the level where they're above threshold, where they reach this trigger zone, we'll get that summation event. We have 3 of these being simultaneously active. And as a result of all of this happening, it will then cause an action potential to be generated in the cell. And so hopefully that makes a little bit of sense to you, and it goes back and reviews some of the concepts that we had in the previous lecture. Another way in which we get sensory cell transmission of information from the receptor to the next cell is where the sensory cell, instead of generating an action potential, will release secretory vesicles when it gets depolarized. And these impulses will then get generated, action potentials will get generated in the next cell, which is typically a neuron. Again, a lot of information. Feel free. Please take a break whenever you can. Put me on pause. I don't mind. You can get me at look at a really weird facial expression. Just put me on pause. Come back when you need to, and if you're ready to go, we'll just proceed. But please feel free to stop at any time. So the second modality is where we get the transmission of these vesicles and the release of these vesicles. So these depolarizing currents that get generated in the receptive sensory cell, they don't produce action potentials. There's no excitable voltage-gated sodium channels at all in this sensory cell. The depolarization that gets generated, though, much like a postsynaptic potential, travels in that membrane. And by the time they reach the end of that cell or parts of that cell, they will depolarize the membrane sufficiently at a level that can actually activate voltage-gated calcium channels and allow for the influx of calcium, which will then be used in exocytosis. So pretty cool. Again, I'm hoping that you remember all of the things that we talked about related to exocytosis and the release of neurotransmitters as well. And this sensory cell is therefore transmitting a signal not through the generation of an action potential directly, but it is releasing vesicles that will then act on the next cell over where it may be able to act on producing an action potential. So let's go through a specific example. Let's look at a taste receptor. So inside of your mouth or on your tongue, you might find these different types of taste receptors that are embedded within these taste buds. And again, we'll get to that later. We'll have an eating party or something, and we'll sample online together when you're watching or when I'm also lecturing. We'll get together a whole list of food to prepare in advance, and we'll eat that together. But when we look at these different types of receptors that are found embedded within this taste cell, and this is that sensory cell, these are going to respond to different chemical stimuli. And oftentimes, depending on the type of tastant or the type of molecule that you are eating, the depolarization that results travels down the length of this cell. So it might begin locally, it spreads a little bit more, and then it spreads even more. And if it were able to reach the other end of the cell at a distance, it may then cause enough of a depolarization to activate voltage-gated calcium channels, which will then open and cause the fusion and release through exocytosis of these neurotransmitters that are found within this sensory cell. And this happens to be a sensory cell that is a taste receptor cell. And as a result of this, it will cause this next neuron to become activated. And here is where we will produce action potentials. This cell up here, it does not produce an action potential, but it is trying to convey the signal to the next neuron over. Again, hopefully that's not too confusing for you. I'm pretty sure that you just need a little bit of time to go over it. You're a really bright class, so I'm hoping that through repetition or through re-listening to this, that most of it will make sense. If you have questions, of course, the Thursdays or the discussion board or emailing me is a great way to do that. So we've looked at these two different modalities for how we get sensory cell transmission of a signal, in many ways parallel to the postsynaptic potential, whether we're directly generating an action potential that has to get to the trigger zone, or we are releasing vesicles and neurotransmitters to then activate the next cell over. Hopefully that's all clear for you. And we're going to actually start thinking a little bit about these same sensory signals in a slightly different way. So here, we're going to start renaming a few things, and we're going to rename them in a way that I'm hoping is logical for you. So I'm going to actually flip back and forth on this screen from the left side where it's just text to the right side where we actually have a little bit of a diagram that may help to explain this in a slightly different way. So let's take a look at this sensory cell. And the sensory cell is here, and the receptors are actually embedded within different layers of tissue, which is what you are seeing here. And we might be able to apply a sensory stimulus. So it might just be touching this particular receptor that's embedded within these different lamellae or these different layers. Those details are not so important. And so let's take a look. We can have a stimulus and we can cause stimulus to be there, and we are touching the receptor, and this is where we start the touching. It stays on there for a little while, and then we remove the stimulus. So hopefully that's clear. And as a result of that stimulus, that we can actually measure and determine that we're going to turn it on, leave it for a few seconds or milliseconds, and then we're going to take it off. And that's what we've seen over here. And we can actually measure the result of how this sensory cell is responding by looking at the receptor potential over here. So at rest, we don't have a lot of activity. It depolarizes, so it's deflecting upwards. And the receptor potential is a lot like the postsynaptic potential. It deflects upwards. And it's not a perfect square. It starts to actually slope down a little bit. And we'll talk about that phenomenon, but that's really not the most important thing that I want you to take away from this. There's depolarization. This depolarization is of sufficient strength to reach the trigger zone. If we were to measure activity down here at one of these different nodes of Ranvier, we would actually see this spiking activity. Remember spiking? Spiking activity, action potentials. Each of these lines, these vertical hatches, represents the action potential that this voltmeter is able to detect. So during the time that we have a stimulus, we are generating a receptor potential, and in turn, this is generating all of this spiking activity that we know requires voltage-gated sodium channels and voltage-gated potassium channels. So this is that receptor potential, and is going to also vary directly in proportion to the intensity of this stimulus. Now, what does that mean? So let's go over here. If you've understood what I just explained over on this part of the diagram, let's go over here. So let's do the same experiment again, but let's vary the intensity of the stimulus. So this means that I'm pressing harder, and you can actually see it as a larger deflection upwards, which is what you see here. You'll also see that we generate a much larger receptor potential. And as a result of a larger receptor potential, do you remember our discussion about spike trains? And do you remember our discussion about being above threshold for longer periods of time and how your body actually recognizes that? Look at what happens to the action potential. So think about it, think about it. I'm gonna ask you a question in a second. What happens to the number of action potentials when we increase the stimulus strength? Okay, so send me that information telepathically. I guess you're also doing it in different periods of time, but I hear a lot of you saying it's increasing the action potential frequency, which is exactly what we see. And so when we talk about the greater the stimulus intensity, the greater the receptor depolarization, which is a graded potential. So it's stepwise, in other words, it's not all or none. And that means that the more transmitter is going to be released, or we generate a higher action potential frequency. And here we're looking at higher action potential frequencies, which we see here. And of course, the greater the depolarization, we also need to hyperpolarize. And we know hyperpolarization is due to what type of channel? If you said voltage-gated potassium, 100% correct. And we need those to be generating these new spikes. And then this will also, again, depend on the relative refractory period and absolute refractory periods of those voltage-gated channels. So hopefully all of that knowledge that you've built up over the last 5 lectures is now helping you understand what's going on here. So after a while, though, what will happen is for this one particular receptor that is responding to this one particular stimulus, you're going to have a maximum effect. No matter how hard you push down, no matter how strong that stimulus is, that one receptor, it just cannot fire any more action potentials because the voltage-gated sodium channels are just not going to be activating fast enough, even with hyperpolarization, which is what that last bullet point was referring to. So if I wanted to know if the stimulus intensity is actually increasing even more, how would I code for that? How does my body know when there's an increase in signal? How do I get that information above this ceiling? So a greater stimulus, which this receptor can't detect. So we would have to actually recruit additional neurons. So as the stimulus intensity increases, we reach a threshold value or a ceiling value for this particular neuron, but it's not just one neuron. We have a number of them nearby. So we would be able to recruit higher threshold sensory neurons. So they haven't been activated yet. They've been silent, but now with increased stimulus intensity, more and more of them would get activated. So here's an example of what I mean by that. So one way that we can code for different types of stimulus, and I've just shown you that on that previous example, is to increase the frequency of those action potentials. And you can go back and revisit that slide, and all of this should make some sense to you. We're increasing that frequency, and I've shown you that before. And now we've reached the ceiling, the absolute ceiling on the maximal discharge. I can't fire any more action potentials for this one receptor. But I also might have nearby another neuron that contains a different receptor, receptor B, or a different group of neurons. And with increasing stimulus strength, so we've maxed things out over here, we haven't really been activating this one yet, but as we have started increasing the stimulus strength and we are now at maximal discharge, this other neuron that's nearby will also now start reacting to that increase in stimulus. So we're recruiting another neuron, and the two signals will be enough to inform the body that we have an increase in signal intensity. I think that's pretty cool, and it's a very elegant solution to how we use single receptors and single groups of and single neurons, and how we can use groups of neurons to actually help understand what the body is experiencing. And so this gets into this question. So how do we actually decode or code for the different modalities of these stimuli? And one of these ways is to have what is known as a labeled line strategy. And that means that activity in one system, no matter what's happening to that system, means that it will only be able to detect that particular stimulus, and that's it. They won't be able to detect anything else. And again, here's an example for you for this labeled line strategy. And we know that this strategy exists. And so think about your eye. Okay, don't think about your eye, because that's just kind of weird. But okay, let's examine the eye. So your eye, actually, if it was punched hard enough, you would actually see sparks of light. And why is that? Because the receptors in your eye are only labeled for one thing, light stimulation. So if it does get stimulated by a different modality, it doesn't actually signal for like increased pressure. It actually signals using that modality. There's no extra light going into your eye as you punch it, but you see flashes of light in your eye. And please don't do that. It's not good and it can actually damage your eye. But if you talk to other people who've had that experience, the force on the eye actually causes photoreceptors in the back to become activated, and you do see or experience flashes of light. And that's because of this labeled line. Those photoreceptors only respond to one stimulus, and the quality of that one stimulus is light. And no matter what the stimulus is, it's only going to recognize it as light. So we code for things using this labeled line strategy. But all of these sensations also have submodalities. So what if we don't want to know that we have like one stimulus and we have a different stimulus acting on that receptor type? How do we actually signal for all of these different subqualities and all of these different features of the sensory system? How do we encode for all of those things? So this labeled line is not a great strategy all the time, because again, like having physical damage to your eye to see light is not a great way for us to determine what information is being processed. And another way to do this, and a better way to do this, is actually what is known as population coding. And here we have a number of different, very specific types of receptors. And instead of relying on information from one receptor, like the photoreceptor, like light normally, but being punched and pressure on that receptor, stimulating it, not a great way to do it, not a great way to code for information about pressure on your eye. How about if we had a number of different receptor types that are coding for that information, and we are able to code by looking at the ratio of the activity of each of those different types of neurons. And again, it's a little confusing now. It'll make a little bit more sense. So I am setting up for a future lecture when we get back into things next week, but it's a really cool phenomenon as well. So for example, we have these three different receptors. And each of these respond to a wide variety of sensory stimuli, and this is the response that they get normally. So the peak response may be way down here for receptor A. Receptor B may be responding at maximal intensity over here, and receptor C hasn't been stimulated by anything so far, so whatever stimulates receptor A doesn't do anything for receptor C. Receptor B, nothing is going on here. And again, this would be like individual responses, but your body doesn't get information from just one receptor type. It will get it from multiple receptors. So imagine, for example, being able to take the information that's coming from multiple sources. So imagine instead of asking for feedback from just like one student all the time, asking for feedback from multiple students, using that information from multiple students. Some of them might have come in early to a lecture, some might have come late, some might have stayed for the entire lecture, and getting their responses to get an idea of what happened on slide number 13. This is sort of what your body is doing, adapting to the sensory space and looking at for any given stimulus, how each of these different receptors is going to be responding. So receptor C will be firing a lot for that particular sensory stimulus. Receptor B will be firing a little bit, and receptor A not at all. But by using all three forms of information from all three receptors and using that population of receptors and the information coming out from there, your body would have a much better understanding of what is going on in the sensory space around it. And again, that's sort of a cool phenomenon that we'll come back and touch on a little bit later. So we've looked at a number of different features for how we encode for stimulus intensity, how we can recruit additional receptor types. Then we started looking at coding for modality, population coding being a very powerful and very, very robust system, and then using the ratio of information coming from a number of different systems. And again, we'll talk about that a little bit more later on. So we're almost at the end, so just bear with me for about 2 or 3 more minutes, and this will sort of wrap up this particular lecture. And then tomorrow we get to talk about, or not tomorrow, but the next lecture, we get to talk about the blood-brain barrier before we get back into the sensory system. When we are talking about sensory neurons, no matter what type of sensory neurons, but typically for skin and typically for vision, as well as for even auditory systems, for each of these sensory neurons, it's going to respond to a particular area of excitation. So on the skin, the receptive field might be a little patch on the back of your hand, and that would represent the receptive field for these sensory neurons. Another group of neurons in another nearby patch would be a totally different group of neurons that are innervating and making up the receptive field for the next patch. So the receptive field for a given sensory neuron is the area or territory or space in which you stimulate that one neuron. So this is one neuron, and in another position, it's a different group of neurons. And this receptive field is always in relation to a specific sensory neuron. And each sensory neuron will have a different receptive field. Now, again, I'll show you this in this particular diagram. So, for example, the receptive field of on your skin, a cutaneous sensory neuron, so this is a skin-specific neuron, is the skin territory that has adequate stimulation that will elicit a response in this particular neuron. And sometimes these receptive fields are larger, and sometimes it can be very, very small, and it depends on how the neurons are innervating that patch of skin. So for example, a receptive field could be very, very large, and by touching anywhere on this large patch of skin, you will be able to stimulate this neuron over here, versus this other patch of skin where we have very, very small receptive fields, and these smaller receptive fields will activate a sensory receptor in a totally different way. And these ideas of the receptive fields are the most important things that I want you to take away from this lecture. It's something that we're going to come back to, so I'm not really emphasizing it now. We're having this discussion. You don't really need to worry too much about this right now for any of the things that are coming up, but keep this in the back of your mind for next week's lectures as we start going through different sensory systems. Receptive fields and the neurons that innervate them, which is the key thing that I'm emphasizing here right now. All right, so we're at the end. I'm going to stop sharing the screen. And I'm hoping that when we get to the last lecture in this week's module, that we're going to be able to have a little bit of a celebration. I know that it's probably been quite the journey for you, and I know that there's a lot of things that have been maybe not as clear for you, or that you need a little bit of time to kind of process and to think a little bit about. I'm also hoping that over the course of this week, or whenever you've been listening to these lectures, that you've been jotting down some notes. We're almost at the end, so we've almost made it. I think we should have a little bit of a celebration tomorrow. And I don't know what we're going to do. We'll kind of do something a little bit different. But I do want you to take good care of your health. These are tough times. And again, I don't get to see you directly, so we're kind of screen to screen and not necessarily face to face. I still really enjoyed interacting with you, even though I'm kind of just imagining it, but I have a pretty good imagination. And I'm also hoping that you're taking time for yourself. Thanks for bearing with me today. I'll see you tomorrow. Take good care of your synapses. Yeah, just try to enjoy neurophysiology, because that's really the ultimate goal that I want for you in this course. All right, so take care, everyone, and hope to see you again in the next episode.